Today, I'm going to explain a new chapter in SAT biology, which is classical genetics. But before explaining, I have to know what's meant by genetics. Simply, genetics is the transfer of hereditary material from the parents to the offsprings. So the genes are transferred from the parents to the offsprings. So genetic variation will be produced in the offsprings. This is simply what's meant by genetics. But before explaining the genetics, we don't have to forget the father of genetics, who is Gregor Mendel. Gregor Mendel is a Styrian monk. Is the father of modern genetics. In the 1850s, he bred garden bees in order to study patterns of heredity. Mendel's work produced three different rules. What are these laws? The first law is the law of dominance. The second law is the law of segregation. The third law is the law of independent assortment. And we will study every one in details. Now, let's start with the first one, which is the law of dominance, or Mendel's first law. Simply, Mendel's first law states that when two organisms, homozygous and pure, look at these two alleles, they are homozygous, they are similar, they are pure. Look at the other one, it's homozygous and pure too. So when two organisms, homozygous and pure, for two offsprings, traits are crossed. What will happen, guys? Look at this crossing. T capital crossed with T small. These organisms are homozygous and pure. Let's try to do the Punnett square to predict the produced offsprings. So after doing this Punnett square, this T capital and T capital, the first two alleles, will be crossed with T small, the second two alleles. Let's cross them together. So T capital with T small will produce T capital, T small. And here too, T capital, T small will produce T capital, T small. T capital, T small will produce T capital, T small. Finally, T capital, T small will be produced. So what do you observe from these produced offsprings? Do you find the produced offsprings are pure or hybrid? Generally, I am observing that all of them are hybrid. So I can say that 100% of the produced offsprings are hybrid. Guys, are they dominant or recessive? Look at the dominant allele. So all of them, two are dominant. So the offsprings will be hybrid and will be in 100% dominant too. If I need to write that what is the genotype, what is the phenotype of the produced offsprings? The genotype, the four offsprings will be T capital T small, while the phenotype 100% or all of them are tall, which is the dominant trait. Okay. I need to see an example. We need to solve a problem about the first law of Mendel, or the law of dominance. Look at this example. Let's read it together. Explain on the basis of genetic principles crossing between a pea plant has yellow seed color, Y capital, and a pea plant that has green seed color. You see that the yellow one is the dominant, and Y small, which is the green seed, is the recessive. It's a pure, it's a homozygous. It's a pure, it's a homozygous. So this what's said by Mendel's first law, or what's stated by Mendel's first law. To see the offsprings, let's do the Punnett square. So in the Punnett square, I'll put the first two alleles for the yellow seed color, and then here I'll put Y small for the green one. Let's do the crossing together. So the first crossing will produce Y capital, Y small, and here will produce Y capital, Y small. Here also will produce Y capital, Y small. Finally here, Y capital, Y small. So, as stated by Mendel's first law, look at all of the offsprings are hybrid and dominant, which is all of them are yellow seed colored. The genotype will be for Y capital, Y small. What about the phenotype? As mentioned before, as I told you, that 100% are yellow, which is the dominant trait. This is simply Mendel's first law. Let's see the law of segregation. What's meant by the law of segregation? Law of segregation, it states that during the formation of gametes, the two traits carried by each parent separate. Let us simplify it by an example. Two pea plants of hybrid smooth seeds, knowing that the smooth seed trait is R capital, which is the dominant trait, and the wrinkled seed R small is the recessive one. So when we make the crossing, we'll find R capital, R small will be crossed with R capital, R small. Both of them are hybrid. Okay. So let's put the gametes in the Punnett square. For the first two alleles will produce R capital, then R capital, R small, R capital, R small, finally R small, R small. So as you see it here, the percentage is 3 to 1. What's 3? What's 1? Look at the smooth seed. The smooth. So the smooth are 3, and the wrinkled is 1. So the percentage will be 75% smooth seed to 1 to 25% wrinkled seed. Okay. This simply the law of segregation, that they segregate from each other. And as mentioned here, when the crossing takes place between these two hybrid alleles, so we'll find the percentage will be 3 to 1 or 75% to 25%. Okay, let's see the third rule. But before that, let's simplify the experiment that achieved by Mendel. Now he got the parents, the tall and the dwarf, and he made cross-fertilization. So he found all of them 100% dominant or 100% dwarf or 100% hybrid. But when he let the first generation to make self-fertilization, as mentioned in the example I told you now, the result will be 3 to 1. 3 tall to 1 short. Okay. The third rule, which is the law of independent assortment. What's meant by the law of independent assortment? It applies when a cross is carried out between two individuals that are hybrid. For two traits on separate chromosomes. Look at here. Two individuals are hybrid for two traits. Look at here, two traits, and here two traits. The round seed and the yellow. Its color is yellow. A green seed, which is wrinkled. So you'll find here in the law of independent assortment that two traits are found on separate chromosomes. Okay. So suppose they are the parents. Let's get the gametes. So I got the gametes from the parents. I'll find it here R capital, Y capital, and here R small, Y small. So the first generation will be R capital, R small, Y capital, Y small. What will happen if you let, or you made if the first generation made a fertilization? Okay, with the first generation crossed with R capital, R small, Y capital, Y small. What will be the result? If you try to complete this square, you will find the result is like that exactly. 9 to 3 to 3 to 1. 9 round and yellow, 3 round and green. Here 3 wrinkled and yellow, 1 wrinkled and green. Let's see it with an example to try to solve it together. Look at this example. A pea plant of tall stem and the green pod. Look at here. Tall stem and green pod. Pollinates another one of short stem and yellow pod. Short stem and yellow pod. Explain on the basis of genetic principles the genetic structure of the first and the second generation. Before, let's get the parents first. Are the T capital, G capital, G capital that will be crossed with T small, G small, G small. Okay, let's get the gametes. The gametes is T capital, G capital, and here T small, G small. Let's get the first generation, which is F1. So it will be T capital, T small, G capital, G small. Now, the first generation will make fertilization. And we need to get the second generation. Okay, which is F2. We'll make it here. Let's complete this table. You'll find the first one, T capital, T capital, G capital, G capital. Second one, T capital, T capital, G capital, G small. The third one is a T capital, T small, G capital, G capital. The fourth one, T capital, T small, G capital, G small, and so on. You can complete the table by yourself. And after completing it, I need to find the percentage of the produced offspring. If you try to get the tall stem and green pod, what is the number of them? I symbolized it for you by this symbol, or this sign. 1, 2, 3, 4, 5, 6, 7, 8, 9. So the number is 9. What about tall stem and yellow pod? 1, 2, 3. What about short stem and green pod? It will be 1, 2, 3. Finally, the short stem and yellow pod, it will be 1, this one only. So the percentage will be 9 to 3 to 3 to 1. It's simply like that. Okay, now we finished the 3 laws of Mendel. Okay, let's see some concepts. What's meant by incomplete dominance? Incomplete dominance, it means no allele is dominant. But what happens? The two alleles blend together, creating a new blended phenotype. How that? Look at here. Did the red flower, did the white flower? I found all of them are pink. So the red is not dominant and the white is not dominant. But if you remember, Mendel said, if this crossing took place, so it will be 100% hybrid. Okay, so, and the dominant. But no, if you find here, no one is dominant. So in this way, I'll find that all the produced ones are pink. And in this case, I can't call it as a Mendelian. So it's called as non-Mendelian. These 4 incomplete dominance. So the incomplete dominance, it's characterized by blending. The two characters blend each other. Let's see other 2 examples from the general life. If a long watermelon crossed with a round one, okay, so if the produced all are oval, it means no one is dominant. If a black animal crossed with a white one, the produced one will be gray. It means no of the two characters, black or white, are dominant. So they blended together, producing the gray one. So this is called incomplete dominance. So since neither trait is dominant, the convention for writing the genes different capital letters. Okay. Let's see this example. When red tulips are crossed with white tulips, all of the offsprings have pink flowers. What percent of the offspring will be red? What will be pink? And if we cross the two pink, what happens if we cross the two pink tulips? Okay. So there, let's symbolize the red one with R and the white one with W. So the pink will be RW. If both of them are crossed, what will happen? Let's complete this Punnett square. I need the percent of the red one, the percent of the white one, and the percent of the pink one. Okay. If we complete it, you will find it. R capital, R capital, R capital, W capital, R capital, W capital, and W capital, W capital. You will find the red is 25%. The white is 25. And the two are pink. So the percentage of the pink one is 50. Okay. This for incomplete dominance. But what's meant by co-dominance? Co, it means that both of them will be shown. If you got these two cows, one is white and the other one is red. So look at this, both colors will be shown. So it's called together. It's called dominance. Both traits will appear. I want to see an example to try to answer it. When two bunnies mate with black, when white bunnies mate with black bunnies, all of the offspring have black and white spots. What type of offspring will be produced if a black bunny and the spotted one are mated? Okay. Let's symbolize with the black as B capital, B capital. Sure, the spotted one will be B capital, B small. Both of them are capital. It's a codominance. Okay, so it's a spotted one. Let's put it here on the Punnett square. So B capital, and here B capital. Let's complete it as I told you. So what will you observe? You will find the black one is 20. The black one here is 50%. And the white one, no white one is 0. And the spotted one will be 50%. Okay. Now, after finishing this part, we need to know some information about multiple allele. It's very easy. It's very simple. Multiple allele, that most genes in a population exist in only two allele forms only. Like the pea plants, it could be either tall, T capital, or dwarf, T small. But when there are more than two allelic forms of a gene, it will be called multiple allele. Exactly like in humans. There are four different blood groups, A, B, AB, and O. They are determined by three alleles, A, B, and O, as mentioned exactly in the figure. A and B are codominant and are often written as IA and IB. What's meant by IA and IB? IBA, this allele expresses the A protein on the RBCs or red blood cells, while the IB, the allele expresses the B protein on the red blood cell, while I small allele expresses neither proteins on RBCs. Okay, so look at here. If I try to crossing I A capital with I A capital and I A with IB, you will find here the percentage as mentioned before, and you'll see here A and B will appear. It's a codominant or codominance. Okay. Finally, what's meant by polygenic inheritance? Generally, many characteristics as skin color or hair color and height result from blending different pairs of alleles. If it resulted from blending different pairs of alleles, it's called polygenic inheritance. Okay. Let's take an example, which is the skin color. The pigment melanin is responsible for dark coloration in skin. There are at least three genes which control for human skin color, where the production of melanin is controlled by contributing alleles, denoted here as A, B, and C, resulting in dark skin color. Light skin color is produced by non-contributing alleles, denoted here as A, B, and C. So the way that the alleles interact is different to those Mendelian genetics. In an example, using two parents heterozygous for each of the melanin producing genes A, A, B, B, C, C, you'll find here, okay, this what's called the polygenic inheritance, that you'll find many, okay, that you will find blending different pairs of alleles. So blending different pairs of alleles is called polygenic inheritance.